Welcome to the Global WarWatch Network. China may be pushing science into some pretty uncharted territory, injecting tardigrade DNA into human cells. Now, could this be the first step toward real-life super soldiers, or is it just science fiction gone maybe a bit too far? Here on The Debate, we're going to be dissecting the profound implications of CRISPR-Cas9. That's the powerful gene editing technology, specifically in the context of this recent research involving tardigrade DNA. We'll explore its potential for, well, enhancing human cellular resilience, and also the very real discussions around possible military applications. All of this framed by, rigorous scientific and ethical analyses.
Right, and the central question guiding our discussion today is really this. Does the genetic engineering of human cells with this kind of extremophile DNA like we're seeing in this research, does it truly represent a legitimate scientific frontier, for therapeutic benefits, for human resilience, or does it dangerously open a path towards ethically questionable super soldier augmentation with potentially unpredictable societal consequences?
Okay, and I'll be arguing that this research, despite some of the more sensationalized ways it's been framed, actually holds significant therapeutic potential. I think it can genuinely enhance human resilience for really critical applications, things like space travel, disease treatment, which makes a strong case for continued, but yes, responsible scientific exploration.
And I'll be arguing to the other side that these kinds of genetic modifications, especially with the looming shadow of military applications, they cross crucial ethical boundaries. They really highlight the severe risks, unintended biological consequences, societal impacts. And that necessitates, much more stringent ethical oversight and a fundamentally cautious approach.
Okay, so to start, let's just appreciate the precision of CRISPR-Cas9. It's a gene editing tool that allows for really targeted modifications, and it's truly revolutionary for biomedicine. Now, the recent research focusing on tardigrades, those incredible water bears, famous for surviving extreme conditions, it really showcases this power. They successfully inserted the Dsup, that's the damage suppressor gene, a unique tardigrade protein, into human HEC293 cells. Those are a common human embryonic kidney cell line used widely in research. Doing that's a remarkable scientific achievement in itself. And this isn't just theoretical. It has, I believe, clear therapeutic and resilience potential. Just look at the evidence. Experiments showed a pretty notable around 40% reduction in X-ray-induced single-strand DNA breaks and also double-strand breaks in these human cells expressing Dsup. It significantly protected their DNA from damage caused by reactive oxygen species. And importantly, this isn't some isolated finding. There was an Italian team back in 2021 reporting that Dsup increased DNA damage repair genes in human kidney cells. And Chris Mason's research in 2020, it suggested Dsup could actually mitigate damage during spaceflight. Mason, by the way, is a known proponent of human genetic engineering specifically for space safety. So these advancements, they hold immense promise for treating debilitating diseases. Think cancer, inherited blindness like Leber congenital amaurosis, cardiovascular conditions like familial hypercholesterolemia, and maybe even more broadly, they could fundamentally improve human survivability in extreme environments.
Okay, I understand the scientific excitement. I really do. But we just cannot ignore how quickly the narrative surrounding this kind of research, regardless of the scientist's original intent, mind you, shifts to things like super soldiers who survive nuclear fallout in the headlines. That immediately throws up a profoundly troubling ethical dimension. It squarely highlights what we call the dual-use dilemma in biotechnology, the potential for a technology to be used for both good and bad purposes. And let's be clear, the concept of genetically engineered super soldiers, it isn't just science fiction anymore. It's being actively explored by militaries around the world. In fact, military policy analysis actually categorizes genetic engineering as an invasive enhancement. And it explicitly warns that these kinds of enhancements, they can lead to, and I quote, very low cohesion when these soldiers try to reintegrate back into an unenhanced society. And we have historical precedents for this kind of thing. Think about the military use of performance-enhancing drugs like Pervitin by German soldiers in World War II. It led to severe side effects, circulatory problems, cognitive disorders, psychiatric issues. It really shows the dangers of prioritizing military necessity over individual well-being and, crucially, genuine consent. Similarly, pyridostigmine bromide, or PB, used in the Gulf War. It caused long-term health problems for many veterans, subjecting soldiers to irreversible genetic modifications without their full, uncoerced consent. That fundamentally violates moral autonomy. It violates Kantian ethics, the principle that you treat individuals as ends in themselves, never just as tools.
That's a very valid concern about the external framing and the historical context, absolutely. But let's bring it back to the actual research for a moment. The Chinese paper itself, it detailed that the Dsup modified human embryonic stem cells, specifically the HESC H9 Dsup line, these are cells valued for their ability to become almost any cell type. They maintained normal cell morphology, normal pluripotency, meaning they kept their developmental flexibility, and they showed a higher proliferation rate without significant apoptosis, which is programmed cell death. Now, that provides some very promising initial safety indicators, at least in these cell lines.
In cell lines, yes.
Precisely, in cell lines. And the research explicitly states that these Dsup-modified cells can be used to study the gene's effects on stem cell development and differentiation, and also to investigate DNA protection. This lays a solid foundation, for clarifying the molecular mechanisms and exploring potential applications down the line. It clearly implies a careful, phased scientific development strategy. And let's remember, many forms of therapeutic gene editing are already considered ethically acceptable, especially when they're aimed at restoring health or curing diseases. Look at the FDA-approved clinical trials for cancers, for sickle cell anemia, for beta thalassemia. This research, I argue, aligns perfectly with that principle of using biotechnology to improve human health.
While those initial safety indicators are, positive, sure, they don't fully address the fundamental unknowns, do they? The Chinese researchers themselves conceded, and this is important, that inserting a tardigrade gene sequence into a human genome may raise safety concerns that need further investigation. That's a crucial admission right there. It tells us there are significant unknowns. We could potentially be talking about severe DNA aggregation and loss of cell vitality from the overexpression of these DNA-binding proteins. Even if that wasn't immediately observed in these initial cell line experiments, it represents a grave long-term risk for actual human application. And, a Shanghai-based scientist expressed concerns quite publicly, saying that the purpose of the gene editing experiment is to change human genes, warning of a kind of game that may not have a happy ending, especially if it's driven by capital or, indeed, military objectives. Essentially, that slippery slope from therapy to enhancement. And crucially, current gene therapy, it's largely confined to somatic cells, non-reproductive cells. It specifically avoids human germline modification because of the irreversible inheritable consequences for future generations and the potential impact on human diversity. Research in human pluripotent stem cells, while not directly germline editing, it definitely pushes us closer to that ethically fraught area. It challenges the principles of using genetics for mercy, not for vanity.
Okay, let's definitely discuss navigating that line, therapy versus enhancement, especially when we're talking about resilience. The Dsup protein's ability to suppress DNA damage by around 40%. That's a precise, measurable protective effect. Now, this isn't about simply creating a physically stronger soldier in the, comic book sense. It's about enhancing fundamental cellular resilience. And that could be absolutely critical for survival in extreme environments, like space, where, frankly, natural human physiology is just insufficient. I see this as akin to providing vaccinations or protective gear. It's a therapeutic measure against harm. That distinction, therapy versus enhancement, it isn't always so clear-cut, is it? Improving resilience against radiation, for example, that can easily be seen as a therapeutic intervention for dangerous conditions, much like how we use CRISPR to treat cancer or genetic diseases. I believe ethical frameworks must evolve to accommodate these beneficial resilience enhancements, especially when we have regulatory bodies like China does, with its strict genetic data laws for collecting and exporting genetic data to ensure responsible oversight.
My colleague raises a good point about evolving frameworks, but the slippery slope from therapy to enhancement, well, it's a well-established concern in bioethics for a very good reason. And this particular research, I think, exemplifies just how difficult it is to control the narrative and the eventual application of such a powerful technology once it's out there, especially when the military context immediately slaps on the super soldier label. While yes, the protective effects of Dsup are noted, applying this to healthy individuals, particularly within a military context, that clearly shifts the paradigm. It moves from treating illness to augmenting capabilities. And that raises profound questions about, well, what really counts as safe and effective given the unknown long-term consequences? Even the FDA's proposal to classify genetic engineering modifications in animals as animal drugs, that underscores the need for strict regulation even outside of human application. It indicates the significant risks perceived in these kinds of alterations. That level of caution should absolutely extend to human interventions.
Right, but when we consider the societal and ethical costs of, let's call it human performance enhancement, military human science does have a responsibility, I think, to explore the art of the possible to meet future challenges. And yes, that naturally includes investigating concepts like the super soldier. However, and this is key, this does not automatically imply reckless implementation. Enhancements can span a really wide range, from non-invasive things like wearable tech and optimized training, all the way to biological interventions. And, if society generally accepts human augmentation, something explored in concepts like transhumanism, then maybe the issue of soldier reintegration into an unenhanced society becomes, well, less severe. The ultimate goal here, surely, is to avoid a binary choice between, say, expendable soldiers or lose the war by carefully developing alternatives and countermeasures. And besides, non-invasive methods like advanced simulations, Jason Dozois talks about this, they're already being employed to develop super soldiers by training cognitive and social skills for complex operations, all without any invasive procedures.
I have to disagree fundamentally with that assessment. Military studies clearly indicate that these invasive enhancements lead to profound issues of cohesion and societal reintegration for soldiers. There's a crucial difference between a non-invasive Iron Man suit, if you will, and an invasive Captain America scenario. Genetic modification is permanent. It's irreversible. That raises the cost-benefit ratio to what I would argue are unacceptable levels for healthy individuals. The ethical imperative to treat people as ends, that means respecting their bodily integrity and their capacity for genuine consent. But military pressure, it often makes truly uncoerced consent, well, nearly impossible. And the potential for creating microtribes, a fragmented society through significant human alteration, that isn't just some abstract philosophical concern. It poses a national security risk in itself by eroding the very social bonds that a military is supposed to protect.
Okay, but regarding the unintended biological consequences, I still believe it's premature to project these worst-case scenarios onto what is essentially foundational research. The Dsup research on human embryonic stem cells, it shows considerable promise for understanding protective mechanisms, and it did so without immediate negative impacts on cell vitality or morphology in those experiments. This is science at its exploratory stage. The broader advancement of biotechnology, including CRISPR, has already led to countless breakthroughs in medicine and agriculture. Even something like gene drives, despite the well-documented concerns surrounding them, they offer potentially powerful tools to restore balance of ecosystems if the appropriate safeguards are developed alongside them. The scientific community, I believe, is committed to transparency and public dialogue to ensure responsible development. The potential for Dsup to contribute to preventing disease or mitigating damage in harsh environments, that's a compelling reason to continue scientific inquiry. Cautiously, yes, but also openly.
I just come at it from a different perspective. The inherent complexity of the human genome, the epigenome, It means that introducing foreign DNA carries unpredictable risks, risks that go far beyond what you might see in initial cell line observations. The source material itself, as we noted, mentions the possibility of severe DNA aggregation with overexpression of these binding proteins, and it highlights the significant work still needed to improve the CRISPR system itself, because of challenges like immune response, delivery complications, things like cell damage, even cell death. Furthermore, this convergence with AI and synthetic biology, that could potentially lead to the development of completely new pathogens, maybe even the recreation of extinct high-risk ones. That vastly amplifies the risk of misuse, whether it's intentional or accidental. This confluence of factors, for me, warrants a much more cautionary approach. It demands increased funding for bioethical research specifically on the long-range impact of germline modification, and a firm resistance to any kind of hasty drive towards a post-human future without thorough societal deliberation first.
To summarize my view, this groundbreaking research into tardigrade DNA, it offers a really compelling glimpse into how we might leverage nature's own extremophiles for human benefit, particularly in areas like cellular resilience and protection against environmental damage. Now, while the ethical considerations are absolutely paramount, they should, in my view, guide, not halt, the responsible exploration of such powerful technologies for their potential therapeutic and protective applications. Ultimately, this kind of work can enhance human well-being and our capability in challenging frontiers that demand these novel solutions.
And to summarize my position, the pursuit of genetic enhancement, especially when it gets linked, almost inevitably it seems, to military applications, and this super soldier narrative, it raises truly fundamental questions about human nature, about autonomy, about societal structure, the risks, unintended biological consequences, the erosion of informed consent, the potential for creating deep societal divisions. These, I believe, far outweigh the perceived benefits at this stage. We absolutely must ensure that the sort of allure of super soldiers doesn't overshadow our commitment to stringent ethical boundaries, to extensive long-term research into the holistic health and, importantly, the cohesion of humanity.
Our discussion today certainly highlights the profound implications of these gene editing advancements. It reminds us, doesn't it, that with extraordinary scientific power comes the responsibility for deep ethical reflection and careful societal dialogue. The material definitely contains much more to explore as we navigate the future of human capabilities. Thank you for joining us today on The Debate. We appreciate your time and participation. Please remember to like, share, and subscribe to Global WarWatch Network.
Yes, and please do share your thoughts in the comment section below. Join us on social media as well. All the links are provided in the description. Thanks for listening.